morning, everyone. I'm Kiew from the Department of Pharmacology. Welcome to today's breakfast at UMHealth live webinar. It is my great pleasure to introduce to you our speaker for today, Dr. Muhammad Farid Nazer Muhammad Faruqu. Dr. Farid pursued his undergraduate and master degree in the area of biochemistry in the University of Cambridge. He then completed his PhD in nanomedicine and postdoctoral research in the King's College London, focusing on the use of exosome for the delivery of anti-cancer drugs and siRNA, as well as for regenerative medicine. He is currently serving as a postdoc in the Department of Pharmacology and will be joining our department in October as a senior lecturer. So the title of his talk today is Exosome, Emerging Star in Nanomedicine. So Dr. Farid, the floor is yours.
Very much, Dr. Kiew, for the introduction. Assalamualaikum warahmatullahi wabarakatuh, and good morning, everyone. I'd like to thank Breakfast at UMHealth as well for the opportunity to share a bit of my research that I've done and some of the plans that will be continue in So today, let me share my screen. So today I'll be sharing a bit about exosomes. What are they and what are their unique characteristics and how that has been beneficial for the applications in nanomedicine. So first, let's understand what exosomes are. So exosomes are basically a type of extracellular vesicles secreted by the cells. So some of you might have come across the term extracellular vesicles before, and they basically mean the entirety of the vesicles secreted by the cells. So roughly, they can be categorized into three different subtypes. So first are the medium or large EVs, or sometimes they are referred to as microvesicles. So these are extracellular vesicles that cells secrete simply by shedding of their plasma membrane. And the second type, the small extracellular vesicles, small EVs, or more commonly known as exosomes. So they're about 50 to 150 nanometers, and they have an endosomal route in terms of their secretion by the cells. And the third category is the apoptotic bodies. So these are just like blebs or fragments of the cells when the cells undergo apoptosis, and they are much bigger in size, of about 1 to 5 micrometer in size. So today I'll just be focusing about the exosomes, or the small EVs, and how they are beneficial in terms of nanomedicine. So let's look a bit more detail on how the exosomes are used. So as I mentioned before, they have an endosomal in terms of the secretion cell. So what this means is that they first form by inward invagination of the plasma membrane to form the early endosomes. And then as the early endosome matures, different biomolecules such as proteins, DNAs, or RNAs starts to get sorted into the exosomes. So when this happens, the membrane of the early endosome will then invaginate inward again to form these tiny vesicles inside them. So as they mature, they will form this structure called multivesicular body. And within them, you will have these intraluminal vesicles. So this multivesicular body, they are either degraded when they fuse with the lysosomes, or if, for instance, the MVB fused with the plasma membrane of the cell, they will then release their intraluminal vesicles, after which, when they are released, these vesicles will then be referred to as exosomes. So some interesting facts about their biogenesis process is that the exosome transmembrane proteins would therefore, given their biogenesis route just now, would mirror that of their parental cells. So whatever is expressed on the parent cells would be found on the exosome membranes as well. And these surface proteins would have the same protein topology as the parent cells as well. So if you look a bit closer on the structure of the exosomes, so they are basically phospholipid bilayer membrane bound vesicles. So you have the phospholipid bilayer membrane on the outside, and then they will have an aqueous core inside. So within the exosomes, or what we call the lumen, are different sorts of biomolecules. So you have different kinds of proteins, as well as different kinds of nucleic acids, such as mRNA, microRNA, non-coding RNAs, as well as DNA as well. And you can see that on their membrane as well, there are different kinds of transmembrane or integral proteins that you can find. And in terms of their lipid composition, since they will have the same composition as that of the plasma membrane of cells, but the exosome membrane are enriched in terms of their cholesterol, ceramide, and sphingomyelin amounts. So because these proteins can be very different from one exosome to another, and cells secrete a lot of different exosomes, there can still be some of them which are conserved, and therefore can be used as distinguishing markers when we want to isolate them from cells. So some of the example of canonical exosome markers that are conserved are the tetraspanins. So they're called tetraspanins because they are transmembrane protein that spans the membrane 4 times. So the conserved ones include CD6, CD3, CD9, and CD81. And there are also components of the escrate, which is in the lumen of the exosome, such as Alex and TSG1. So there are other negative markers as well, so proteins that should not be present in the exosome, such as connexin, or other kinds of proteins which are ER or associated, as well as lipoproteins. So these should be derived from the plasma. So these are one of the ways how we can distinguish the exosomes from other kinds of extracellular vesicles secreted by the cells. So a bit of the history on how exosomes were discovered. So they were first observed by Penn and Johnston in 1985. So they were actually studying the maturation of sheep reticulocytes and how the cells get rid of their transferrin receptors as they mature. So first, they see that using electron microscope, they can see that the cells have this multivesicular body structure that I described before, and it contains the intraluminal vesicles. And they see that as this fused with the plasma membrane, it will then release the intraluminal vesicles. So initially, they thought that this is just a means of a waste disposal of getting rid of the transferrin receptors. But the transferrin receptors are the ones that are stained with the immunogo antibodies, so these black dots that you see. But then later on, so they are then only 2 years after their first observation, the term exosome was coined. So basically, exosome means body which is outside the cell. So only about a decade later that they sort of like revisited exosomes, and these were initially discovered by Raposo et al. In 1996. So she found that these exosomes were also secreted by B cells, and they were found that these exosomes carry peptide-loaded MHC class II molecules. So from then on, then they realized that they actually play a role in antigen distribution from one cell to another. So from here, then they started to see the exosome actually have distinct roles in physiology, and from then, exosomes have been discovered to be secreted by almost virtually any cell types that you can find in the body. So where can we find the exosomes? So current practice, or the most conventional method of getting the exosomes, are from culture, cell culture. So you can either use culture cell lines or primary cells. And then how we collect them is that we culture these cells, and then we leave them for about 2 to 3 days. And then once we collect this culture medium supernatant, or in the exosome field, they are mostly referred to as conditioned medium, or CM. So when we get this culture medium, and then we do some isolation steps, and then we can obtain the exosomes. So exosomes can also be found in all sorts of physiological fluids. So it has been shown that they can be found in serum or plasma, in saliva, in urine, in the CSF, breast milk, and even in semen. So, and more recently, they have been found to be secreted by plant cells as well. So we can find them in cell culture of plant cells, fruit juices, and even mesh tissues of the plants. So given their physiological abundance, so what roles do they actually play in the body? So we can basically look at exosomes as the physiological version of our post-Laju or GNT. So what they do is basically all the biomolecules that they encapsulate within their lumen, they transfer these proteins or nucleic acids from one cell to another. And then upon delivery, these biomolecules can then elicit function in the recipient cell. So here is an example. So a donor cell, for example, would secrete the exosomes with all the different biocargo in them. So when delivered into the recipient cell, all these biomolecules was then to start to elicit their function, and then would result in a functional outcome in the recipient cell. So basically, when more work has been done, it has been, so people started to see that exosomes actually is a form of intercellular cell communication. So previously, it was thought that hormones or cytokines are the major mediators of intercellular communication, especially distant ones. But then a lot of work has been done to see that, actually exosomes are also the main mediators of intercellular communication. So here are some examples of the roles of exosomes in mediating normal physiology. So as mentioned before, in the field of immunology is where the boom of exosome function was first studied. So they were discovered that they deliver antigen, they help with antigen presentation as well as distribution in the different antigen presenting cells. And they also play a role in immune suppression. So Tregs, or T regulatory cells, secrete exosomes containing immunosuppressive biomolecules. In the nervous system, they have been shown to mediate neural outgrowth and survival. They involve damage repair as well as neurotransmitter homeostasis. And in the cardiovascular system, they stimulate endothelial progression, migration, and tube formation, as well as help in optimizing glucose metabolism. So, of course, if they play a role in normal physiology, they will also play a role in pathological physiology. And of course, cancer is the main disease that people have realized the role of exosomes in mediating cancer. So they promote tumorigenesis by delivering all the tumor-promoting molecules. They help prepare pre-metastatic niche preparation according to the seed and soil hypothesis. So the cancer cells from the primary tumor site can release exosomes, which are then released and then lodged in the metastatic site and prepare the site for metastasis. They also help in mediating drug efflux from the cancer cells. So if we incubate cancer cells with cancer drugs, we can see that the exosomes secreted by the cancer cells would have this drug containing them as well. They also play a role in therapy neutralization. So for example, HER2 breast cancer cells, they release these HER2 positive expressing exosomes, and they are the ones that mediate neutralization of the trastuzumab, so the HER2 antibodies. And therefore render the therapy ineffective. And they also play a role in immune evasion, again, by secreting exosomes with immunosuppressive properties. So in terms of neurodegenerative diseases, exosomes have been implicated in mediating the spread of pathogenic protein aggregates. So for example, in Parkinson's disease, they help transmit alpha-synuclein. In Alzheimer's disease, they help transfer the A-beta protein as well as the delivery of prions from one cell to another. And also in infectious diseases, so people have also discovered that not only eukaryotic cells secrete exosomes, but parasites as well as bacteria also secretes some sort of extracellular vesicles. So these can be either exosomes or sometimes it is referred to these outer membrane vesicles, and they play a role in the regulating the host pathogen interaction. So this could either be the delivery of virulence factors or similar to the cancer, they can also help with immune evasion from the immune cells. Now, in terms of nanomedicine, exosomes have been used mainly for these four purposes. So mostly they are used for drug carriers. They have also started to gain interest to be used as cell-free therapeutics, as diagnostic biomarkers, as well as for vaccine development. So today I'll just be focusing on the use of exosomes, some example for the use of exosomes in drug chronocarriers, cell-free therapeutics, as well as a short example on their use as diagnostic biomarkers. So in terms of exosomes for drug delivery, so similar to other nanocarriers that's been used for drug delivery, exosomes offer the same advantages, such as physical protection. So the drugs, when we encapsulate them, they will be protected from degradation in the plasma, for example, even delivery. The carrier is also versatile in terms of they can deliver different kinds of drugs. So you can deliver small molecules, or you can also deliver biologics such as siRNA or therapeutic proteins or peptides, given their natural role in delivering these biomolecules. They can also deliver both hydrophilic and hydrophobic drugs. So hydrophilic drugs will be contained in the aqueous lumen, while hydrophobic drugs will usually intercalate within the membranes. So in terms of biocompatibility, where the exosomes play a big advantage, given their physiological abundance, they are non-immunogenic. But however, this is still tunable. So if, for example, you isolate the exosomes from the immune cells, especially the antigen-presenting cells, such as the dendritic cells, they can be immunogenic and raise an immune response. But however, if you derive it from other cells, which are not involved in raising an immune response, then they will be non-immunogenic. So they are biocompatible with tunable immunogenicity. Another advantage is that given their biogenesis and they inherently load biomolecules in them, there is potential or the ability of the exosomes for have these drugs, for example, biologics and even small molecules to be endogenously loaded in them, so that when we isolate these exosomes from cells, they are ready to use nanocarriers loaded with the drug. So you don't have to do this post-isolation loading anymore. And you can also engineer the exosomes for active targeting. So you can see that the exosomes have surface proteins or transmembrane proteins. And if you create fusion proteins of these proteins with some active targeting ligand that can target the exosome to certain tissues, then you can have exosomes with active targeting properties. But another sort of like distinguishing property of the exosome is that they are reported to have this intrinsic homing property. So what this means is that some people have found that exosomes from different cell types tend to home to certain tissues of interest rather than so they have this sort of like intrinsic homing pattern, and mostly they have been shown that they tend to, especially in the context of cancer, they tend to be taken up by the parental cells compared to other cells. This is something that I'd like to share an example with in my previous work. So here, so we started to look into the homing property of exosomes ourselves. So what we have is that we isolate exosomes from this PANC1 cell. So this is a pancreatic cancer cell line. And then we administer these PANC1 exosomes into a mouse bearing either the PANC1 tumor, the parent cells, or B16F10 tumors. So this is another tumor. So we just have a mite bearing two tumors, and then we see whether how these two tumors behave in terms of taking up the exosome. So what we see is in vivo. You can see here in the left panel, the top one. So the PANC1 tumors, we can see that over time they steadily take up exosomes, whereas with the B16 tumors, they take up the PANC1 exosome, but not in a similar manner as the PANC1 tumors. And then when we dissect the tumors and we measure the exosome amount in them by fluorescence, so we fluorescently label the exosomes, we can see that the PANC1 tumors significantly take up more exosomes compared to B16F10 tumors. So in this context, for example, we were able to see that the exosomes do have a preferential accumulation into their parental cells, which is the PANC1 tumors. So having seen this, we then decided to do some therapy work. So we have the PANC1 exosomes loaded with this siRNA that's targeting PAC4. So PAC4 is one of the oncogenes that have been implicated in pancreatic cancer proliferation as well as migration. So we use electroporation to encapsulate this siPAC4 into the exosomes. And then in vitro, so we first tried, so we use the exosomes containing the siPAC4, and we compare it with lipofectamine. So lipofectamine is a conventional transfection agent for nucleic acids. And we can see that the exosomes are more efficient in resulting the knockdown of the PAC4 expression compared to the lipofectamine. So then the same dose of siRNA was administered in both cases. And in terms of migration, we can also see that, so if you see in the last two pictures on the right, so again, here we can see because of their more efficient knockdown, the exosipAC4 is also able to induce or suppress migration of the cancer cells compared to the lipofectamine SIPAC4. So in vivo, then we have the same mice bearing the PANC1 tumors, and we administer these exosomes into them. So first, we can see that in 9 days, upon administration, so with the control, so exosome administered with the SINEC. So this is siRNA that is just random unscrambled. So we can see that the exosome loaded with the SIPAC4 is effective in suppressing the tumor growth. And this has therefore then resulted in a significantly improvement of the survival of mice post-implantation of tumors. So then that's where our interest in developing the exosomes as drug delivery nanocarrier increased. And so we decided to further understand their biodistribution in vivo. So this is work done in collaboration with AstraZeneca. So we tried to sort of like establish the best way to track the exosome in a sensitive, accurate, and reliable manner. So we compared 3 different imaging modalities, the fluorescence, radioactivity, as well as bioluminescence. And we label the exosomes in the different ways to reflect, so that we are able to track them using these different modalities in vivo, and then we do the biodistribution analysis. So this is the result. So the labeling method using the for nuclear imaging is what I developed during my PhD, and AstraZeneca decided to sort of like expand and do this comparison to understand their biodistribution studies. So next, we'll have a bit of a look into exosomes as cell-free therapeutics, and this is mainly done in the field of regenerative medicine. So a bit of the definition of what regenerative medicine means is referring to the repair, replacement, or regeneration of cells, tissues, or organs to restore impaired function resulting from any cause. So although we probably won't reach the level of Mr. Wolverine here, having instantaneous regeneration, but hopefully it can, the field of regenerative medicine is aiming to improve and accelerate the repair of dysfunctional tissues. So for a number of years, people have known that the MSC or mesenchymal stem cells have this regenerative effects, and they have been, so rather than initially people think the MSC exert the regenerative effects by differentiating into the tissue that they are being administered to, the regenerative effects are actually achieved by paracrine signaling. So the MSC secretes an array of substances called the secretome, and from here we get and this is what's actually mediating most of the regenerative effects seen, such as proliferation, anti-inflammation, anti-apoptosis, as well as angiogenesis. So in terms of exosomes for regenerative medicine, so people have discovered when people analyze the secretome of the mesenchymal stem cells, so lo and behold, they found these vesicular structures, which is about 130 nanometers and contain these markers. So basically what they found is that exosomes are also part of the secretome, and they are also playing a role in terms of exerting the regenerative effects observed. So since then, people have started looking at MSC-derived exosomes for regenerative therapy in different injuries or disorders of tissues such as kidney, heart, liver, and as well as brain. So the use of exosomes rather than the cells themselves is what cell-free regenerative therapy is about. And they have these certain advantages compared to the cell-based therapy. So first, we can avoid aberrant situ differentiation of the MSC, the stem cells. And then we can also avoid lung embolism, because that's what happens when we intravenously deliver cells. And then the exosomes, again, as I mentioned, they have lower immunogenicity compared to cells. And they can also, so although the exosomes themselves already have the therapeutic agents, we can further load them with other drugs that can further improve the regenerative effects that we want to see. So a bit of our work before exploring the therapeutic properties of the umbilical cord-derived mesenchymal stem cells derived exosomes. So we first do a 3D culture of the cells in this special plate called aggregate plates. So they have this like conic bottoms that allow the cells to aggregate and form the spheroids. So this is how the UCMSC, the umbilical cord-derived mesenchymal stem cells, look like when they have formed the spheroids. And from here, then we isolate the exosomes from these spheroids. So the reason we culture them as spheroids first is that because there have been reports showing that 3D culture of the stem cells compared to 2D culture can help them show better regenerative effects. So once we get this exosomes derived from the UCMSC spheroids, we treat them on 3T3 murine fibroblasts. And what we can see is that we can see an increased migration of the fibroblasts. And this is a key step in the wound healing process. And not only that, what we can see is that if we treat the cells with the UCMSC spheroid-derived exosome, we can also see an increase in the proliferation of the fibroblasts. So again, this is also an important step in mediating the wound healing process. Okay, and last, I'll just touch briefly on the application of exosomes as diagnostic biomarkers, and this is mostly done in the terms in the context of cancer. So here I show an example of how this might work. So people initially done work on HER2 positive breast cancer cells. They found that the exosomes secreted by these HER2 positive cells are also positive for the HER2 expression on the exosome membrane. And when people do further study, when they isolate when they collect plasma or serum from HER2 breast cancer patient, they can also see that the serums or plasma samples are enriched with the HER2 positive exosomes. And therefore, if we, then this basically highlights the potential of using exosomes as diagnostic biomarkers. So as I mentioned before, the membrane protein expression of the exosomes will mirror that of the parent cells. So this will be useful in terms of exosomes because some of the oncogenic proteins are transmembrane proteins. So in terms of using exosomes as diagnostic biomarkers, not just surface proteins can be used. People have shown that some nucleic acids in the exosome, especially microRNA, can be used as the diagnostic biomarkers, as well as some other luminal proteins that can also be differentiating markers between exosomes derived from healthy individuals compared to cancer individuals. So this then leads to the use of exosomes as diagnostic biomarkers will rely heavily on biomarker discovery work, and this is mostly done using proteomics to really comprehensively profile the difference in terms of these contents of these biomolecules between healthy and cancer individuals. And also, it would need to rely on the development of biosensors with high specificity and sensitivity for detecting them. So basically, the advantages of using exosomes as biomarkers is that these biomolecules, given that they are either encapsulated in the lumen of the exosomes or spanning the membrane, they are better protected from degradation when they're in the circulation. So that's why that's the main advantage of using exosomes. And also in terms of detection, you can create biosensors that captures the exosome first using the conserved markers such as the tetraspanins, and then you can clear away all the debris and other proteins of non-interest in the plasma before you do the detection. So that can help with specificity and sensitivity as well. So again, although it sounds amazing, exosomes do come with challenges. That's why not many of them has been approved for clinical applications yet. So the major issue with exosome is basically the heterogeneity. Because every cell makes exosome of slightly different composition. So even the same cell will make different exosomes. No, exosomes are basically like snowflakes. No two exosomes are exactly identical to each other. And another point is that they are heavily influenced by culture conditions. So this is a double-edged sword, actually. So it can be beneficial in terms of when people reported, for example, stem cells, when you culture them in hypoxic condition, the exosomes derived from them exert better anti-hypoxic effects, but this is not necessarily true for other conditions. So it can be that you lose a therapeutic effect when you change the culture condition. And then in terms of the content of the exosome, so because if we use them for drug delivery, we don't necessarily empty the content, more like we're not able to at this moment. So in terms of cancer-derived exosomes, so we know that they mediate tumor progression and metastasis. So if we use cancer-derived exosomes for drug delivery, what is the effect? Can we still see the therapeutic effect, or will the content, the original content of the exosome sort of like nullify the therapy that we want to achieve? So there has been efforts to empty the content of exosomes for use as drug delivery, and these are called nanoghosts. But then this has been extremely difficult, and is still not successful in terms of conserving the integrity of the exosomes when trying to empty its content. Also, a huge debate in the extracellular vesicle community is that which EV or extravesical population are we isolating? So we might think that we isolate exosomes, but at the moment there is no definitive biomarker or marker that can differentiate between the different extracellular vesicle subtypes, as I mentioned earlier before. So that's why it is suggested that we don't really use the term exosome, but rather just refer to them as extracellular vesicles, although we see the conserved markers that I described before. So the other two issues are, of course, the purity and the yield. So, for example, in a good setting in the lab, we can get about 10 to the power of 12 particles of exosomes. Whereas we compare that to other synthetic nanocarriers, such as liposomes, one preparation can easily get you about 2 orders of magnitude higher. And then there's also the problem of which EFD subtype I mentioned before. And also in terms of purity, when we isolate them using current methods such as ultracentrifugation or size exclusion chromatography, the samples might not be actually completely devoid of non-exosomal vesicle and other proteins as well. So, and that would rely heavily on the isolation methods used. In terms of scaling up production, because we would need to use cell culture to obtain large amount of exosomes. So that's the challenge in achieving the economy of scale as well as GMP production. And because cell culture is involved, that's why the cost is also quite high still and needs optimization in terms of the production. So nevertheless, we can see in the past 5 to 10 years, there's a boom in exosome mediated therapeutics. And these are examples of certain companies that which basically have exosome as the central technology in achieving therapy. So there are about 50 plus companies as of now, and some of them are involved in making vaccines for COVID-19 as well. So I'm sure they are working on some ways to get the approval from FDA, for example, in terms of the challenges I described before. So basically, in terms of what I plan to continue in is basically to explore the niche of exosomes for biologics delivery. I have them endogenously loaded with the drugs, and hopefully create designer nanomedicine. So in terms of we selectively incorporate the active pharmaceutical ingredient as well as the active targeting in this case. And I hope to explore this first in cancer in regenerative medicine, and hopefully to explore them in biomarkers for cancer screening as well as for use in antibiotic resistant breakers as well. So this is, if you want to delve into exosome research, you can visit the International Society for Extracellular Vesicles. They have their own journal, which of just a very high impact factor from 11 to 25. And this is a very important paper if you want to understand further about the minimum requirement for the study of extracellular vesicles. So these are just my collaborators from my previous lab who has helped me a lot in establishing my expertise in exosome studies. So with that, I thank you very much for your attention, and happy to take questions.
Okay, thank you, Dr. Farid, for the interesting presentation. We will now just read out one question. The rest maybe we'll answer it on online through typing. Okay, so one burning question from Prof. Tunku Sarah. She said, I'm an orthopedic surgeon. What do you think, how exosome could help in bone healing and treating osteoporosis?
Right. So that's a very good question. I have not come across studies using exosomes for use in bone disease or bone disorder related therapies. But I'm sure, for example, in your case, the use of exosomes derived from stem cells would be beneficial. So it depends on what therapeutic effect that you want to observe. Is it like mineralization, restoration in the bones, or increased proliferation of the osteoblast compared to osteoclast, for example? But then I would assume that it could be the exosomes from these cells, for example, the osteoblasts, would help in mediating the effects that you see. So, and I come across actually in Nazer, they have started to use platelet-derived extracellular vesicles as well. So these are also another source of extracellular vesicles with regenerative and therapeutic properties. So that's perhaps something that could be explored as well.
Okay. Okay, for the interest of time, I think we will stop this session here. We thank Dr. Farid for the interesting presentation. So Dr. Farid will answer the rest of the questions online later. Okay, so now we will pass the time to Prof. Azlina to introduce the second speaker. Thank you. Over to you, Prof. Azlina.
Thank you, Prof. Kiew. Assalamualaikum and salam sejahtera. Hi, I'm Azlina from the Department of Molecular Medicine. I will be the moderator for this second session. So we are now moving from exosomes and nanomedicine into a different topic on snake venom. So please allow me to introduce the next speaker, Dr. Kae Yi. Dr. Kae Yi is a senior lecturer based at the Protein and Interactomics Laboratory in the Department of Molecular Medicine. He is our homegrown talent, having obtained his PhD from He is an upcoming researcher, and his research interest covers various aspects of toxinology. His work in snake venom is part of a research field that was started more than 30 years ago in our department. So Dr. Tan has taken the work further by utilizing new technologies, which include application of omics technologies in studying venomous snakes, antigenicity of venom toxins, and snakebite diagnostics. So his research team works closely with local and international experts, and he has an extensive list of collaborators, including from Thailand, Pakistan, Taiwan, and Italy. So can I please now invite Dr. Kae Yi to present his talk on snake venom, deadly toxins of life-saving therapeutics.
Okay, thank you very much. Good morning, everyone. Thank you very much, Prof. Azlina, for the very kind introductions. I hope you can hear me well. Okay, today I would like to take this as a sharing session because snake venoms look pretty mysterious to many of people. And in Faculty of Medicine, University Malaya here, we are actually looking to the snake venoms, which is from the deadly toxin translated into life-saving therapeutics. In this talk, I would basically divide in 3 parts. Before I go into talk about the more technical aspect on the research, I would like to give a very brief introduction into the snakes and also venoms, as well as the consequences of snake bite. So to many people, snakes is perceived as evil because it causes destruction, causes death. But for some people, they think that it's positive sign or symbol that represent rebirth, transformations or healing, as what we can see in the role of Eclipus, the Greek god of medicine, where you can see is holding a rod here and a representation of perceived as the medicine logo, as what we can see in the WHO symbols. So in general, I think everyone knows that snakes is long, slanted in body shape. It comes with scales, either rough or smooth. Can come with many colors, many sizes, but of course, not all are actually venomous. So for those venomous snakes, it basically comes with pairs of highly specialized delivering mechanisms that basically help them to inject the snake venoms from the snake venom glands into their prey. And this snake venom is actually located on both sides of the head, slightly behind the eyes, that the snake venoms that being injected is mainly serves the functions for preying, hunting, or defense. So you might be thinking that snake venoms is often injected when they bite human, but actually they are not, because in order to produce the snake venom is metabolically expensive. So especially when they are not preying on human. So I would like to introduce also the word poison, because these 2 words has been interchangeably used in the earlier days. Some people still refer snake venoms as poison, but definitely they are not, and has been well clarified. So I'm giving you example here. So if, let's say, you bite something and you die, it's considered as poisonous, such as the mushroom, like the frog, poisonous frog, or the poison pufferfish. If they bite you and you die, it's considered as venomous. From the example would be the snakes, the spiders, or bees. So it's just as simple as that in classified that. And all the snakes, although they look alike, but down there, genetically, they are actually different. And thus, they are actually producing a very diverse types of venom toxins to form the cocktails. So basically, in a snake venoms, it can have up to 100 types of toxins. But from our studies, we managed to figure out there are around 15. They are from 15 major protein families, and they can be diverse and vary across different genus, species, or sometimes even within species, they might have slightly difference in the compositions. So looking into that, we can know that the pharmacological actions that it can induce is actually very diverse, and hence causing various toxicities upon the bite. I would like to take a look on the consequences of snake bite. So in snake venoms, basically, it can generally classify into the various types of toxins. For neurotoxins, it's mainly targeting the neuromuscular junctions to disrupt the signal transmission between your synaptic junction. For hemotoxins, it basically attack different types of factors or platelets in your blood coagulation system to cause hemostatic derangement, whereas for cytotoxic, it can cause tissue damage. But this tissue damage, if not being treated well, it can cause necrosis and subsequently may lead to amputation to save the rest of the parts of the tissue. So this is what will happen if, let's say, it's not treated properly. For venomous snakes, it basically classified into two different major groups. Can be a Viperidae or Lapidae. So basically, most of people, they don't know how to classify them. But for the Viperids on the left, as I think many people would think that the venomous snakes or the snakes with triangular heads would be venomous, and this is these types of snakes, venomous snakes. Whereas on the right, you hardly differentiate them except for cobra, because the rest of the species they basically have a rounded in shape, and sometimes it will be mistakenly identified as non-venomous snakes. So in general, from pathophysiology, we can see that from the left, they are likely to elicit hemotoxicity, whereas the venomous snakes from the right, they are likely to elicit neurotoxicity. But sometimes it's just a general classification, but a combination of toxicities might occur in some snake bite, because the snake venoms are very diverse, and a different group of snakes, they are likely to have combinations of two types of toxins. Today, snake bite, I would like to share the distributions of the epidemiology, and we can see that Asia remained the most impacted regions. This is mainly because and also affecting the regions and also populations that mainly engage in agriculture or fishery activities. When we look at the statistic, we can see that the death caused by snake bite is actually very high, and much more than many other neglected tropical diseases, and has been referred as bigger public health crisis by Kofi Annan, the former Secretary General of the United Nations. But what is not worth to mention is that the victims of snake bite is actually being left and also experiencing a very neglected way to die, because antivenom is not easily available in many countries. So, and this further emphasized the importance of the research to actually translating the deadly toxins into life-saving therapeutics, especially the snakebite antivenom treatment, which is very much needed nowadays. Besides that, through the understanding of the snake venom's compositions, we can develop them into therapeutic drugs or diagnostic applications, which I will elaborate further later. So snakebite treatment, if maybe some of you do not know, that is the definite treatment for snakebite and still being conventionally produced. It's being produced through the injection of a small amount of snake venoms into the bigger animals such as horse, sheep, sometimes camel, depends on availability. And we harvest the blood, get the immunoglobulin out, and being packaged as an antivenoms in different forms. And of course, the specificity of these antivenoms would be specific for each different type of species. In reality, although we think that the specific antivenoms is very much important, but in reality, they are not available and very much limited supplies. And especially, these are the image of the antivenoms that are available in the regions. And in Malaysia, we don't have antivenoms being produced, but we are highly rely on our neighboring country, Thailand. But we are very lucky that they have the best series of antivenom ever. As you can see, it comes with different colors. It indicates different, specific again, different species. So this is mainly due to the sharing of venomous snake species across the two countries, likely because of the ecological similarity. Okay. So what we do here in University of Malaya is looking into, of course, the snake venom's composition, as well as to look into how effective is the antivenoms in neutralizing the toxicity that induced by the venom preclinically, because we can't do that in the clinical settings. So taking this as a very simple example that we have published some time ago. So this species, Nazer cobra venoms that sourced from 3 different countries, is being profiled with different types of approach. So here is the chromatogram. We can see that it's very is substantially different between 3 species. When we look into these fractions that distinctively present in the NKT, which is from Thailand sample, we are very ambiguous that what is that, and we further identify them using mass spectrometry and found out that it's actually a very highly toxic components that can elicit neurotoxicity. But these fractions definitely is not actually found in the other 2 species, which consistent with our finding showing that the venom is highly lethal with very low LD50. So when we look into the neutralization to explain on how the Thai antivenoms work on our Malaysian samples, we see that they're actually having a comparable efficacy. So what we can explain from here is that the antivenom is being produced by from these Thai samples, which likely to have similar protein components except this one. So they are able to neutralize it, although it consists of these very little toxin, because the antivenom produced are likely to be similar. So, however, if you try to put the scenario the other way around, it might not work, because the antivenoms, if let's say is produced from the Malaysian samples, it may not contain this lethal antibody that's again these lethal fractions, and the toxin that injected into the victims will not be successfully neutralized. And this is why I say that the clinical research on looking into the efficacy and paraspecificity of antivenoms is very much important. Just now, we only take a look into only 1 species, but there are many cobra species out there. Not only that, there are many group of snakes that worth investigating too, because of the current limitation and also choice of antivenoms. So I want to take you to another side, another challenges of the snake bite, which is we are actually facing the very important issues that we are having a very poor efficacy of antivenoms, and this is likely because some of the venoms contain the principal lethal toxin, which is the causative toxins that is low in antigenicity, and hence leading to the production of a low antibody titer that can against these principal toxins. This further indicates or reflects that a high dose of antivenom is needed that increase the risk of hypersensitivity of anaphylactic shock in the patient. So what we do here in University of Malaya, together with the Thai collaborator, we are trying to come out innovative treatment preclinically through identifying the principal toxins and enrich them in venom immunogens during an immunization, and hope that the cocktails will give rise to the antibodies that are able to neutralize a wide range of venom species which has similar properties. Other than that, we also collaborate with other institutions to look into other innovative treatments such as single chain variable fragment, or the inhibitors that can inhibit the action of the snake venom proteins to aid the snake bite management. Next, I would like to take you further into the other topics, which is therapeutic drugs derived from snake venom proteins. So the most popular one being the captopril and ACE inhibitors that are used for treating hypertension. On the left here is the tirfibant and also eptifibatide acts as an antithrombotic, and also the betrosorbent anticoagulant. And this anticoagulant has been developed into the pellet gel to aid the healing of the lesion or the wound in a surgical and mainly for surgical use. And these are all clinically available nowadays, and many of them are actually undergoing clinical and preclinical studies, which involve in the which mainly targeting the coagulant cascade, but some are actually work as anti-cancer, mitogenic, or analgesic agents. In our labs, while we are looking into the venom compositions, we also found out that there are various types of bioactive components which may serve biomedical potentials. However, the discovery of new therapeutic drugs is a long journey. Not only require the collaborations from different parties, but importantly is the continuous research funding, which is very hard to achieve in the academic settings. Okay, I'm so sorry for that. Doesn't really, I'm so sorry. Doesn't work well. Okay. Okay, next, I would like to bring attention to the diagnostic applications, because as I mentioned earlier, the snake venoms contains a very diverse and non-enzymatic or enzymatic toxins. So using these properties, we can develop them into the assay kit, which are able to detect or screen abnormalities as an indication of the disorders. In our lab, we are interested on the medical healthcare sides, which is the diagnostic applications for the identification of snake venoms, and also to actually aid the timely administrations of antivenoms. We are currently working on that. We hope that with this timely administration, we are able to improve the treatment outcome, because the earlier the interventions, the earlier the neutralization of the toxicity in the patient. Not only that, with the diagnostic kit available, we are able to know what species biting, which can aid the management of snake bite and also distributions of the antivenoms to the regions that it needs. On the right here, the image right here is actually the snake venom detection kit. The only one in the world is from Australia. Why we are actually lack of diagnostic kit in clinical setting is because snake venom toxin are actually very diverse and complex. So they do have some sharing protein in between the different species, that making it challenging to develop a very accurate diagnosis. So in our labs, we are trying to use our experience in snake venom research, finding out those unique toxins that can actually which can actually produce the antibody against it for the specific detections, and hope that it can be implemented, but it's still in the research, in the progress of research, and we are likely to gather more information before the publication. In summary, like many other research, snake venom studies are going to a stage that requiring interdisciplinary or transdisciplinary research to bring its impact. And this is because we ourselves alone cannot be actually doing everything, and we need to collaborate with different parties. So I welcome those or anyone that interested in snake bite or snake venoms to actually collaborate with us. And this is because it's not only limited to the scientific research, but also social science research, such as the management of snake bites, healthcare, and also the educational programs carries equivalent impact. So it is finally, towards the end of my talk, I would like to share with you some take-home message. Snakes look alike, but not all are venomous. So don't simply kill them if they are not harmful to you, because they are important parts of ecology systems. And the second one, if you are so unlucky to be bitten by one, go to the hospital immediately and get a specific treatment, because many people, I think most of people, they can't identify which one is venomous and which one is non-venomous. And thirdly, and the final one will be the universal antivenoms. I think many people are hoping to have one, but honestly, we don't. So this is why research has to be carried out in order to get one that near to the universal antivenoms. But eventually, because of diversity of snake venoms, we hardly to do that. So this is the Detox Lab research team, and currently headed by Dr. Tan Chuos from Department of Pharmacology. Those standing behind are postgraduate students. And on the left here is our honorary professor who has experience in snake venom research for almost 40 years and has built a solid foundation for us to continue his legends. And I would like to also thank Prof. Sim Simwe from Department of Pharmacology, which has been involved in snake venom research with us for quite some time as well. Okay, that's the end of my presentations. I thank you very much for your attention.
Very much, Dr. Tan Kae Yi, for that very insightful sharing. Just to share with you the Q&A session, there's a question from Prof. Tukusara. You know how earlier you were saying about what to do when you get bitten by a snake? So she was asking, should we take photo of the snake that bit you?
Yeah, yes, of course. If you are bitten by the snakes, the first thing you have to do is to stay calm. It's hard, but you have to, because if not, then you will expedite the blood circulation and the toxin will distribute faster than you think. Okay, so next, if you are able to take a photo, that please do it, because it will actually help the clinicians to identify them to give you a specific treatment. If not, then probably someone around you can kill the snakes, bring the snake specimen over to the hospital so that they can identify it for you. If really not, then you have to wait. If let's say there's envenomation or the venom is actually injected by the snakes, there's signs and symptoms that will develop, and clinician will, based on this syndromic development, and decide which antivenom to be administered. But this is just true guessing based on experience. And this is why diagnostic tool, as I said, is also part of the important part of the research that we are going to look into it.
Thank you. There's another question from Dr. Farid. He's asking, can you vaccinate against snake venoms? Is identified from common venomous bite people?
Yes, technically, yes, because if you inject snake venoms, then you will produce antibody. So there are very many species of snake venoms that snake species that probably you have to try every of them in order to just have a little antibody to against a large amount of venom being injected into your body. So it's not practical at the end of the day. This is why we are still relying on the antibodies that based from the animals in order to treat the patients or the victims.
Maybe I can ask just one last question. This has something general. How you mentioned earlier that there's very limited supplies, right, of this antivenom, especially the ones that's produced from horses. Why is that so? What's the reason for the limitation, Dr. Tan?
Okay. Regarding this question, actually in Malaysia we have the anti-venom production facility at 1970 somethings, but it is being shut down because it costs a lot, millions per year, and this is the most important reason for that. Another reason would be there are too many species that we can't cover every species in order to just provide a specific treatment, because if we do so, that will be needing a lot of money, a lot of horses, a lot of equipment, just to produce, to fill the demand of that. Of course, in Malaysia, we don't have many venomous envenomation cases compared to probably Thailand, Indonesia, or Philippines, because we do have thousands of cases per year, but it's still manageable through the antivenoms being imported from Thailand.
Well, maybe one last question, since we are coming to the end. Professor is asking, do you think the big pharma element is keeping it expensive?
Yes, pardon?
Is there a big pharma element that some the company keeping it expensive?
I think it's not, because there are a few pharma companies actually producing antivenom earlier, but they cease production because they don't get any profit from it. And most of the antivenom manufacturers nowadays in various countries actually are subsidized by the government, because or not, it will be shutting down, and the poor people will be suffering, because snake bite mainly affecting a poor population engaged in the agriculture activities. So, and it's mainly paid by government anyway. And we normal people, we don't buy antivenoms usually. So they don't likely to make up the prices. But one antivenom from Australia, I'm not sure about that, because it costs 25,000 ringgit per vial. And in an in a case of, that's the systemic antivenoms. In a case, then probably victims may need 2 to 3 vials to get a full recovery. So it's really costing a lot, which this is why we are proposing the wide spectrum antivenoms to fill the needs.
Thank you so much, Dr. Tan. Since we're now
thank you.
And I see there are one more question, but perhaps I leave it to you to type answer in the chat box later. Thank you, everyone. So should you have further questions later, please do reach out to Dr. Kae Yi at the Department of Molecular Medicine. With that, I would like to thank everyone for joining this morning's session of Breakfast at UMHealth, and I hope this was beneficial to you. Do have a fruitful day ahead. Assalamualaikum and salam sejahtera. Thank you.